So far we have covered that there is a G1 phase where we are prepping all of the materials, an S phase, which is synthesis, where we are making all the things that you would need in a second cell, G2 phase, where you're organizing it and getting ready for mitosis, where you actually undergo the cell separation. We have a few checkpoints. We have the G1S checkpoint, then we have the pre-S or S checkpoint, and then we have the G2m checkpoint. And these three checkpoints are what we're going to be looking at. How do these checkpoints work? That's kind of what this entire PowerPoint is about, is how do we get past and what is happening in each of the checkpoints. So, in the G1S checkpoint, where we are wanting to begin to put materials together and get ready for synthesizing everything, we are going to have, you guys have seen the retinoblastoma protein, the RB protein before. And so we're going to see that again. So here's DNA. And sitting on top of the DNA, we have the E2F protein that can't do anything right now, because right on top of the E2F protein we have the RB protein that looks kind of like a half moon in your slides. And the RB protein is keeping the E2F site closed, and so there can be no transcription of making all that stuff. And so this, the RB protein is named after retinoblastoma. It's a type of cancer, and that's because if this is broken, then the E2F can move forward when it shouldn't, and that'll cause cancer, because this is the beginning of cell replication. And what is cancer? Uncontrolled growth of the cell. So just pulling that together here. So here we are going to have a cyclin-dependent kinase. Cyclin-dependent kinase come in, and this cyclin-dependent kinase, its name for this one is coming up soon. I believe it's just named after the phase, but I want to make sure it doesn't have a special one. So yeah, just G1S CDK. So G1/S cyclin-dependent kinase comes in and will phosphorylate the ends of the retinoblastoma protein. And so then this will look like this, where E2F is still there and will move forward with making a whole bunch of things, because retinoblastoma protein has been phosphorylated, which are these dots on the end, which takes it off. And that is what this first checkpoint looks like when you zoom in, is you just send in a cyclin-dependent kinase, and then that will phosphorylate the retinoblastoma protein and allow you to have a whole bunch of cell products. So how could you see this in the final? You could see it based on which phase is this in. When do you see the retinoblastoma being acted on? When do we care about it? It's first checkpoint. G1S. You could see it, I believe that she visited retinoblastoma as a disease, as cancer. And so she could ask, oh, someone has a retinoblastoma. She probably wouldn't use the name. She might try and describe it. What proto-oncogene might be messed with, or what protein might be broken? And so it would be the retinoblastoma gene or protein. So those are things that might come up. All right. Any questions? Yes?
So the retinoblastoma, it's a disease and it's also a gene?
Yes. So we saw the disease first, and we named the disease, and then we found out what caused the disease was a certain gene being broken. And so we called it the retinoblastoma gene and the retinoblastoma protein.
So by broken you mean it's supposed to be attached to the G2m
Almost all the time.
all the time so that there could be cell division manipulation. But when it's
Right.
dephosphorylated,
So when
it comes off with E2 and then that's how you get the cells to keep multiplying and
when it is malformed so that it can't hold on to the E2F, no matter what the phosphorylation going on is, it's just, it can't ever cover it up. And so it floats around useless, and the E2F is always being activated. And so therefore, cancer. And so this would be, a retinoblastoma would be which? A proto-oncogene or a tumor suppressor gene? Think about it for a second. That could be a question, and so it's good to understand. Tumor suppressor, yes, definitely. And that means how many copies need to be broken for you to get cancer? Both copies or only one copy? Because you have
wait, it's recessive.
back there, 2. It's recessive, exactly. And that's because if you make some broken retinoblastoma protein and you make some fixed, correct retinoblastoma protein, then the ones that work will go ahead and do the job, even if the other half are floating around useless. It still can suppress it moving forward. So even if you only have one good copy, you're okay. Tumor suppressor ones, they are considered recessive because if you have one good copy, it can still do the job, typically. So this one is a tumor suppressor, and you need 2. All right? Make sense? And that's that one I actually would be totally unsurprised if that was on the tests, that kind of a question, because that's the kind that I seem to recall she likes. All right. Any other questions? Okay. Cool deal. Let's go ahead and see if she went into the synthesis phase. She kind of went into the G2m first, but I'm trying to go in order. Okay, well, I'm going to go ahead and go to the G2m because so far I'm not seeing her talking about the S checkpoint, and I would want to slide on that one. But G2m. So G2m, we're going to have a G2m Cyclin-Dependent Kinase. Okay, and it has 3 spots on it for phosphorylation where you can stick phosphates.
What's the name of that molecule?
G2m CDK. Also, it can be called the mitotic, which I think is what you guys have in your slides. Actually, is mitotic CDK. So mitotic cyclin-dependent kinase. Okay, so it has 3 spots. Now, typically it floats around like this. Most of the time, this is the checkpoint being closed. It has 2 phosphates in its inhibitory sites. So that's what those eyes right there I'm putting are just, that's the inhibitor site. You will have phosphates in both inhibitor sites. It floats around, it does nothing, which is good because we don't want the cell to divide unless we want the cell to divide. Otherwise, it's called cancer. And you guys are going to get your fill of cancer coming up soon. Now, when we do want the cell to divide, we're going to have a kinase that we don't need to know the exact name of come in and phosphorylate this activation site in the back. So it's going to stick that in, and sticking it in on the back is going to pop these 2 phosphates. Not directly. It's going to ask for a phosphatase. We don't need to know the exact name of the phosphatase, but it is an enzyme, and this being here allows the phosphatase to come and then knock both of these out. Make sense? So basically, stick that in, pop those 2 guys out, and then this will no longer have those phosphates in, and it will tell this phosphatase, good job, go do that more. And so this phosphatase will be upregulated and be like, okay, gonna do this even more. And so it's a positive feedback. Go free all of my brethren. All right. And then we have an uprising. And then we make a movie, and then we're rich. All right.
Yes.
Next.
Your brain.
So I'm just going to go ahead and show this cyclin-dependent kinase, like so, with its little phosphate on the back here. And this mitotic CDK is going to go ahead and activate 2 more enzymes. It's going to activate laminin, which I'm going to draw like that, and it's going to activate condensin. Laminin, condensin. All right, so laminin, or laminase, I should have said. I believe it's laminase. There we go. Laminase is going to affect the membrane, the nucleus. And so when we're going to do cell division, we need to open the nucleus up so that we can have access to the genetic material and put it between the 2 cells that we're going to be creating. If the nucleus is still there, we can't do that. And so laminase is going to go ahead and start breaking up the nucleus. Okay? So it kind of dissolves away. Condensin is going to go in and make so that all the DNA is condensed. And so it goes from the free-floating squid-looking thing to the little X's that you typically see when we're trying to show it in a cell division state. Okay? So
Taylor,
yeah?
Are you saying that the laminase breaks down the nuclear envelope?
Yes.
Okay.
Exactly.
And it condenses the chromosomes.
Yes. And so it's getting those, both of those make sense for when you're having cell division, that you would want those to happen. Nuclear envelope is in the way. Chromosomes have to be condensed in order to be pulled apart and separated without being a bunch of streamers that can be broken easily. Okay. So, part 1, part 2, part 3 is going to be moving to where our mitotic CDK is going to turn on ubiquinin tagging. Ubiquinin tagging. Who knows what ubiquinin tags do?
Isn't that where it tags the cell for either destruction or, well, no, ubiquitins is destruction.
Excellent. Exactly. And so you tag all the things that you want to be broken down. So ubiquinin tagging, it has a second name as well, which is the something or another anaphase promoting complex. There we go. This is also called anaphase promoting complex. Okay? So mitotic CDK is going to turn on the ubiquinin tagging or anaphase promoting complex, and these are going to break down a couple proteins that need to be broken down for cell division to happen, for anaphase in particular. So if we look at a chromosome, in the center it is being held together, and some of the molecules that are holding it together are called cohesin. Okay, because it's coherent, it's holding together, cohesive. And thankfully, we have an enzyme that is always present called separin. And separin will go and chew up cohesin, except most of the time it is being held on a leash by an inhibitor molecule called securin. So securin is going to secure separin and keep it from separating things when it shouldn't. And this anaphase promoting complex will go and tell securin, just let go go ahead and let it happen. Separin comes out, and it immediately will go and chew up cohesin, which is going to allow the chromosomes to be pulled apart by the spindles. The anaphase promoting complex and ubiquitin tagging is going to occur to multiple other products, but this is the one that she was focusing on, and so this is the one that I would know well. Okay, any questions?
What's that word again? After condensin?
After condensin,
The blue section on top? On top?
this?
Yeah.
Laminase.
Laminase.
All right. Can't read my writing. Okay. So, and this is all contained in the G2m checkpoint. All right. And so this would be happening between metaphase and anaphase. So this is metaphase stuff up here. And this is anaphase, prepping for anaphase down
And
here.
that part is the G1S checkpoint.
Yes. Excuse me. This?
This part,
Yeah, this is. And then that's G2m. All right. So, with that, let's take a look. Okay. With the retinoblastoma and G1S CDK, let's go ahead and actually expand this story a little bit because she bounced back to it. And gonna go ahead and get rid of this really quick. So we have a pathway that is called the Rass Pathway, okay? Rass protein. And the Rass protein, when it is activated, is going to go and turn on this cyclin, the G1s cyclin-dependent kinase that'll take off the retinoblastoma and allow it to move forward. So if the Rass protein is going to have that signal, let me ask you first, is the Rass protein, the gene, let's just say the Rass gene, back up a little bit, is this going to be a proto-oncogene, meaning it only takes one to jack up the cell, or is this going to be a tumor suppressor gene? Its job, when it's functioning correctly, is to tell this thing when to take retinoblastoma off or not.
So it's not a suppressor then.
Right. It is not a suppressor. So you only need one bad gene of the Rass protein to tell it to take retinoblastoma off, because as soon as there's one guy telling it to come off, it doesn't matter if the other gene is working properly. You only have to have one jacked up to have the whole process go forward. Now, the Rass protein is usually off based on having a bound, is that a D, GDP? GDP. And as long as that is held in there, then it is inactive. And so when it gets a new GTP from, let's take a look. I don't know if she expects you to remember the name guanine nucleotide exchange factor, but GEF. GEF is the protein that will exchange the GDP for a GTP. I think, if I recall correctly, what she wants you to be familiar with is GDP means inactive.
Yes.
And then GTP means active. She will also want you to know that The GEF is what helps make the GDP go to GTP, but it is the Rass itself that is going to do auto phosphate. It's going to cut off. It's going to go ahead and it's not phosphorylate, auto dephosphorylate the GTP, and so the Rass protein is going to go ahead and act on itself to turn itself off and make this into a GDP. And I'm going to go ahead and make the GTP a little bit clearer that it's different. So GTP, that is on. So this is the part that if it is broken, if it cannot stop itself and it cannot take off a phosphate from the GTP, then that is what causes the Rass to keep going, is because it stays in the active state. And she may ask you, I seem to actually recall one on that, active, is that it's not able to cut off its own phosphate. And so then it's always active. As soon as it is turned on once, it is on forever. Yeah, because it... All right.
Because it's losing its auto ability to turn off?
Yeah, and the part of the Rass protein has a specific name. It is GTPase activating protein, but that is part of the Rass. So GTPase activity. He's probably all she really wants. GTPase. Okay. All right. And then all of these lead to a path of proteins called the MEK proteins, which I remember I memorized the entire pathway of MEK proteins and everything that led down to this cyclin-dependent kinase. And then I don't recall using that particular knowledge. And so MEK proteins are the pathway.
It's a pathway to what?
So Rass will go ahead and act on some MEK proteins. Those will act on others. There's MEK, ERK, Mike, and a bunch of other things. And they eventually go down and turn on the CDK, G1s CDK, and then that'll do the phosphorylation of the retinoblastoma, which frees E2F to go ahead and make a bunch of cell products. So it's just the cascade. It's the protein cascade.
So Rass activates components in the pathway of MEK proteins? Or
Yes. So Rass will turn on the cascade of MEK proteins. RAF, MEK, ERK, Mike. I think that.
My other Uncle Mike.
Sure, you're right. All right. So
Are all of these ways to just answer?
So it's real. The reason cancer keeps coming up is because we're talking about how the cell divides. Cancer is about dividing when we don't want to. So anytime we have cancer, it's because something's broken in the normal pathways of cell division. So we see pretty much all the cancer issues while we look at the normal cell division issues.
I don't want to divert us off topic too much, but is there anything naturopathically we could do to get the GDP, like say in a patient with a mutation, or with the Rass has one gene, to get the GDP to inactivate it or whatever? Is there anything that...
So for this exact pathway and that exact molecule, I don't have anything that I can say.
Yeah.
I don't know of anything that targets that specifically.
Yeah.
There have been some supplements and dietary changes that have been shown to support the cell's own self-catch mechanisms. So we have double check mechanisms of whether this should be happening or not. And if you support those, then there are some, I've heard, I haven't read them myself, but I have heard of some research that shows that cancer, the number of errors in a cell goes down, which I would suppose also means that there's less likely going to be a risk of cancer. So, and that's how much I've heard about it from a presenter. So, good question though. All right. Let's see.
I like that train of thought because we're often like we stop at what we hear, but really we need to think like, okay, well, what caused this, and then like what caused that part that caused this, and then what caused, you know, you gotta keep on going until you actually find a naturopathic mechanism that you can put in control.
Yeah, and your project was on the mitochondria and cancer processes, and then I remember it was quarter one.
Yeah, for the ketogenic,
Ketogenic diet.
apoptosis mechanism.
Yeah. That was so interesting.
Yeah, it was really cool and very helpful for us as well.
All right. Okay. So, welcome, Pedro. Next, we're going to go ahead and talk about the P21 family. So I'm going to go ahead and erase this so I can get a little bit of room. And the P21 family is a family of inhibitor proteins. So, P21, there are several of them, and we are going to talk about, looks like P16. Oh, P16 and P21 families. All right. And CDK4 and CDK6.
Was the MAP, MAPKS, and the E2F, which you're talking about, that you memorized the whole thing? And it wasn't.
All the MAP and the MEC and the ERK and all of
Yeah.
that pathway, knowing, I would know probably like that MAP and MEC are part of this, like how can you know what pathway is being talked about, but I
Transition.
wouldn't memorize the order of all of the
Okay.
protein cascade.
Okay, thank you.
Yeah. So, and also in this, these guys have another name, which is also CDK4 and CDK6 are both part of this. And that's the name that you see on this slide with the P21 and P16. And with that, these guys are the inhibitors that are making so that these guys aren't always doing their job. Okay? And so it's like,
Okay.
hold on to you until we get told to go, which is through this pathway. And so I remember last time, anyway, never mind. So I want to reorganize all the slides, but I didn't have enough time to do that. So with this, 21 and 16. 16 is going to go ahead and hold on to CDK4 and 6, and so that's going back to the Rass pathway, so you can fit them in. And so these are CDK4 and 6. And then P21 is an inhibitor for a lot of the other ones, and that's why it's included. So the rest.
P53 activates it. She loves P53.
It inhibits those? Is that what you're saying?
Yeah, that's the thing that holds on and makes so that they're inhibited.
Okay.
Some of them are inhibited by phosphorylation. Some of them are inhibited because they're bound to one of the P family proteins, P21, P16, P19, what have you.
Can you repeat that?
Sure. So the P family of proteins will hold on to a lot of cyclin-dependent kinases when they aren't needed, so that they don't act on anything. When they are told to let go via some kind of protein pathway, whether it's this one or another one, then they'll let go, and then the cyclin-dependent kinases can be activated and moved forward. And then they'll do whatever their particular job is. In this one, the CDK4 and 6, also known as G1s CDK, will go ahead and phosphorylate retinoblastoma and let cell products be made. Cool? All right. So that's
And the P21 gets activated by P53, right?
That sounds right.
And MGen.
I can't say quite yet.
That's okay.
Didn't get to that one before.
She told us P53 was important for the test.
Yeah, P53 is one of your tumor suppressor genes, I believe, and if that guy gets broken on both, then he's one of the most common causes of cancer is P53, and so you will want to know that I think P53 has to do with apoptosis, and basically P53 is kind of the DNA self-check protein.
And the glycolytic enzymes, it has a part with that.
Yeah, so apoptosis is when it's going to go ahead and chew up everything, so you're going to have a lot of lytic proteins activated, and P53 is the one that says whether or not that cell death happens.
The terminator.
All right, exactly. Okay,
I have a question.
sure.
So when P53 decides to kill
Mm-hmm.
how is that a mechanism?
So you do need to know some of the mechanism. So the cell will typically be receiving survival factors. And these are just proteins that are released from various sources that you're not expected to know, but through the body. They're kind of like paracrine or hormones that are sent. And if the cell continues receiving its survival factors, it continues to operate fine. As soon as those disappear, the cell will wait a little bit, and then it will kill itself. And so if you got rid of survival factors, the cell will die. That would be a really terrible disease. I wonder if there's one like that. I'm a bacteria that chews up survival factors. That's not very good for the bacteria then, because it kills the hosts. That's probably why it hasn't happened. So, go out in a blaze of glory. All right. So, yeah, survival factors is one. The other is...
So I don't know if it would be blaze. I think that would just be like, you just rot.
Right, you're right.
Yeah.
Bacteria they're always thinking
Well,
of
I'm
themselves.
just saying.
I'll need to come up with a better metaphor.
Yeah.
They're always
I'm just
selfish.
helping
They're
out.
always selfish and thinking of themselves. You know, they don't want to do
Yeah.
They
suicidal. They're not.
I appreciate that about them. I like win-win, or at least win, not die. All right. So there's also, I don't see how it says that it, but if you have DNA damage that is confirmed, that's the other way that you can have P53 say, all right, let's let the cell die. So if either of those two things happen, survival factors go away, or there is DNA damage that is confirmed by P53, which is a complicated process, I would guess, since I don't recall learning about it and we just left it at that. Both of those will make cascades to apoptosis. And actually, let's go ahead and just dive into those cascades, because those are ones that you will want to know. How many of you guys have seen DuckTales?
Oh, yeah.
All right,
Oh,
excellent.
yes. I forgot all about that.
DuckTales, woo! All right, so B for my pitch, but DuckTales. Oh, sorry, partying here. So DuckTales is a cartoon where it's just a cartoon that's kind of a comedy cartoon where there's little duck. There's a city full of ducks, and they all do things, and there's Uncle Scrooge who's rich, and he has really weird things that happen to him all the time because everyone wants his money.
Is Donald Duck part of it?
Yeah, Donald Duck is part of it. So there you go. Anyway, there is a nanny in DuckTales named Mrs. Beakley. That's why I bring this up, and that's because that's how I remember this next protein. It is BCL2, so Beacle2. And I'm going to make this cell bigger because we're going to learn some of the proteins involved in apoptosis, since we had a good question taking us that way. So this cell is now oblong. All right. So we
So we're talking about P53, right? Is
Yes,
that where the survival factor comes from?
P53
Okay. I just wanna make
is,
sure.
yes, you're right.
Okay.
P53 is the factor that will allow the chain of events that I'm about to say happen. So P53 usually stops all of this from happening unless it's found cell damage and then it's like DNA damage, then it's lets it just move forward. So you have BCL2 or Mrs. Beakley 2. And BCL2 is normally inhibiting this pathway. There's a bunch of proteins that you don't need to know the names right here. Just call them apoptosis proteins. Okay? So apoptosis proteins are ready to do stuff, but BCL2 doesn't let that happen. P53 says, okay, BCL2, go ahead and stop working, and we're just going to let these apoptosis proteins go through. And after that, you will have procaspase is one that I remember she cared about. So procaspase 9 is going to go ahead and join with, which other ones were important? I think procaspase was the main one that she wanted. There are some other ones listed like cytochrome C and APAF1, but I think that she mostly wanted procaspase because that's a family. Caspase. The procaspase family is involved in apoptosis. And so if you see the word procaspase, you should think, cell death, cell killing itself. So BCL2 is no longer stopping these proteins from turning on procaspase proteins, which will lead to apoptosis.
Okay, so can you review that concept right there?
Yes.
So DNA is damaged.
Yep.
P53
says
if it's
BCL2,
damaged,
go
then
ahead and let
needs
it.
to be
Right.
dealt with, go through apoptosis, right?
Mm-hmm.
So what is BCL?
So BCL2, so P53 and BCL2 both take their hands off, basically.
And let it go forward.
And let it move forward,
Okay.
because it always has these cell death proteins floating around. And these proteins are a whole cascade, I would guess, which is why they're just death proteins. And those will turn on procaspase, which are the ones that actually do the work of killing the cell. And so it's always ready, it's always on a hair trigger, because if your cell starts getting mutated, you don't want to be like, oh, well, with a mutated cell, maybe we can build all the things we need to turn ourselves off. We might not be able to do that, so we always have them ready.
Okay.
And then we have let go, P53, let go, BCL2, and then it
Cell
just
death.
goes in cell death.
Okay.
I don't know if I understood wrong, but is procaspase inactive, and then when it comes to caspase, is inactive?
Yes, and that's the pro part. Thank you for saying that. So the pro means it is the pro enzyme, the before it
Precursor.
is active enzyme. So anytime the precursor, exactly. Anytime you see an enzyme with pro, that means it's before it's actually turned on. So procaspase is when it's in the safe state. Caspase is when it's going to be active. We typically call them procaspase enzymes just because that's how they normally look. But once they're activated, you would take away the pro for sure.
So when it's inactive, that's when it results in apoptosis?
Caspase would be the one that's doing it. Procaspase, it gets turned on to become caspase, and then that chews up the cell.
Okay, so procaspase activates caspase.
Becomes. It's the same thing.
And caspase is the one that does the apoptosis.
It is a major part of it.
Okay. Right.
All right. So, yep. And then if you don't have a survival signal, then for that, really, I wouldn't memorize. Well, let's see. Would she? Did she? For DNA damage, caspase 9. For survival factors, caspase 3 and or 8. So this basically goes straight to the procaspase to caspase.
The survival factor goes to procaspase?
Yeah, if you stop getting survival factors, then the cell will just go straight to procaspase and caspase. It doesn't need to mess with any of the other BCL2 stuff or whatever. It goes straight there if it doesn't get survival factors.
Are we talking about a normal cell here or a cancerous cell? This is a bad cell.
This is a cell that has been deemed worthy of death. So it might be cancer, it might be...
Might you just
It's been
normal cell death? Yeah.
Yeah. So down here is because of DNA damage, it needs to die. Up here is because of some reason the body says it needs to die. It might be working perfectly fine, but no more survival factors means you don't get to survive anymore. And then survival factors are gone means procaspase to caspase apoptosis.
Does caspase is it related at all to capsaicin that's in cayenne pepper?
Capsaicin? No, they're not related. And then these ones are 3 and 8 for the caspase. Caspase 3 and 8, caspase 9 down here. Okay? And that's apoptosis. So, concept question. And it was kind of asked earlier, but you might have been taking notes. Does the cell have to have DNA damage for apoptosis?
No.
No. Okay. If you had cancer, which pathway would you expect to probably be the one that's going to be activated? P53. Why? You're right. Because DNA damage is what causes cancer most of the time. It's either damage or mutation. And so for cancer, you'd expect the P53 BCL2 procaspase pathway to be what we're looking at.
Okay, so that can be environmental, epigenetic, or genetic mutation. Like
Anything that makes so that the
Okay.
DNA is acting abnormally enough that the cell's like, I'm not normal, I need to die. So anything that's outside of normal parameters to grow a lot.
Okay, thank you.
All right. Any other questions? I would know the clinical correlates, the cancer stuff, because that's, number one, a good bank for questions, and number two, you're going to see it coming back in Gen Med, where you're going to have some cancer stuff coming up. And if your cancer terminology is strong, it'll help you.
Awesome, thank you.
All right, so yeah, it looks like we're about at time. Do we have any other questions before we close up?
Can you just explain to me what's going on here really quickly?
Yeah, sure. So a quick review right here. We have the Rass pathway, which in a normal cell first. Normal cell wants to divide, so we turn on the Rass by giving it a GTP instead of a GDP. GTP means it's on, it will do the MEC and the MAP and a whole bunch of other proteins that you don't need to memorize the order, but they're MEC, MAP, ERK, and MIC, and I think there's one other. And those will go down and turn on CDK4 and CDK6, which can also be called G1s CDK, because that's the part of the cell cycle. So you have the G1 phase and the S phase, and there's a little block right here. And this checkpoint is what we're getting past. And so turning these on moves us past that checkpoint because it will come down. The CDK4 and 6 will come and phosphorylate the retinoblastoma protein, which takes it off of the E2F. And that lets E2F go ahead and make a whole bunch of cell products that you will eventually need for cell division. In cancer, cancer, you can have the Rass protein be broken because the gene is mutated. And that means the Rass protein does not know how to turn itself off, because normally it would turn itself back to GDP after it's done cell division. In mutated Rass protein, the GTP stays forever, and it just stays here.
So continually dividing.
Yes, so it always is going to dividing, and that is a proto-oncogene.
Okay.
Because it only needs one broken for it to do this. Down here, the retinoblastoma can be broken, but if it has to have both of them broken. Because if you have some broken that are floating over here and some that are working, doing their job, stopping the cell from using the E2F sometimes, then it'll stop. Doesn't matter that half of them aren't there, whatever, we have enough, we can stick them on there. We'll just use the good ones and make twice as much of everything. And so we just run it more. And so this one is a tumor suppressor, and you'd need 2 copies broken to get cancer. But those are the 2 cancer places. 1 copy, 2 copies. And if you think about it and make sure it makes sense why 1 copy and 2 copies, that'll serve you well, because there will be questions on tumor suppressors and proto-oncogenes.
Oh,
So,
okay, so know the difference.
yeah,
Okay.
which one needs one, might only be 1 or 2 questions, but know the difference between, like, which one needs 1 copy, which one needs 2, and if it makes sense why, then you don't
You
have
have to
to,
work harder to suppress a gene than you do to cause it.
yeah. So,
Good.
that's a good way to think about it. All right.
I have another analogy that might help.
Sure.
The tumor suppressors, like if you think of a car that's always like on the gas and on the brake,
Mm-hmm.
you take off the tumor suppressor and now you just go straight forward. So that's what causes the division. Because it's always on, but it's like stopped by the tumor suppressors. So taking them off is just going to make them just go through cell division.
Exactly, and you would need both of them to come off. So you have 2 feet on the brakes. You take 1 foot off the brakes. You still have brakes. You have to take both feet off. There you go.
Got it.
I have a question about Rass. So if we know this pathway, would that help with the cell signaling? Because I know this pathway was also in our cell signaling lecture.
Mm.
It must be important. And the Jack Strat or Sprat.
Yeah,
Or,
Jack Stat.
Still don't
That's
understand.
actually Jack Sprat. Will eat no fat. His wife will eat no lean. That's actually how I remembered that thing too.
Someone told me they want to name their dog Jack Stat.
Yay, medical students. Good time.
Just feed them a whole bunch of fatty ones.
Okay.
So yeah, this is one of the pathways, and it
Because
just
that
happens
lecture
to be.
scared me, to be honest.
Yeah?
So, yeah.
The questions on it aren't super detailed bad.
Okay.
I don't remember the exact test questions, but
Yeah.
I remember that if you get the concept, you'll be good.
Okay.
Jack,
G proteins and
Yeah.
what is it?
which ones are G, which ones are kinase, and go through that.
Okay.
So-
Okay, and then what about the recombinant lecture? I was sick and I actually missed the whole lecture, and looking back at it, I'm like, and then she's like, well, just know the clinical uses
right.
and correlates for it, but okay.
If you can say, let's see, first there is the,
And she said basically what we know in micro for PCR is all we need to know, but
yes, that's the
okay.
one that you would want to know, PCR.
Which part is that?
recombinant lecture.
And it's the one with the blotting, if you remember that. If you can describe PCR and the steps of PCR, that's the one that I know in detail.
Okay.
PCR and then sequencing and blotting, they all kind of go together. They're all part of the
Okay.
same thing.
She said I don't have to know the different types of blotting for this
That's
test.
nice.
Yeah.
For the rest of the things that were in there, 'cause she talked a little bit about like satellites and things like that, get concepts.
Okay.
So you can find videos on PCR that'll
Okay.
just say, okay, here are the 4 stages of PCR.
Okay.
First, denaturation,
Annealing
etc.
and- Okay.
And then if you
Uh-huh.
get that, then you can find something on sequencing.
Yeah.
Because those are widely talked about. So
Okay.
it'll be easy to find resources on them. So
Thank
don't let
you.
it get too much to you.
Okay. I'm feeling better. I've been this test.
It'll be good. It'll be
Yeah.
all right. Okay, so with that, any last questions?
How bad
Okay.
is mine?
For some reason, these topics are not intuitive.
Good luck.